Good
Good
morning,
morning, ladies and
gentlemen.
Welcome to the opening day of our virtual international conference on biodiversity and biotechnology for food security and sustainability, hosted by Graduate School and Faculty of Mathematics and Natural Science, Universitas Pajajaran. My name is Aji Saputra and my partner.
Raisi Youngest.
It is our great honor to serve as your Masters of Ceremonies on this remarkable occasion.
The virtual international conference on biodiversity and biotechnology for food security and sustainability is a platform that brings together scientists, researchers, practitioners, and policymakers from around the world to discuss and share knowledge on the critical roles of biodiversity and biotechnology in achieving food security and promoting sustainability.
This conference consists of 2 days meetings. On the first day, there will be an opening ceremony, plenary session, and parallel session. Meanwhile, on the second day, conference include plenary session, announcement for the best presentation, and closing ceremony.
Ladies and gentlemen, we would like to extend a warm welcome to our distinguished guests and attendees. The Honorable Professor Doctor in Medicine, Dr. Setiawan, the Dean of Graduate School, Universitas Pajajaran.
The Honorable Professor Dr. Iman Rahayu, MSI, the Dean of Faculty of Mathematics and Natural Science, Universitas Pajajaran.
The honorable keynote speakers, Professor Jyoti Prakash and Professor Johan Iskandar. And plenary session 1, Dr. Namrata Tamang, Professor Dr. Ibnu Dwi Buwono, and Professor Hitoshi Iwahashi. Not to forget to mention plenary session 2, Professor Suseno, Dr. Abdullah Bilal, and Dr. Wan Abdul Al-Qadir Iman Wan Mohtar.
The sponsors of our activities today from Yayasan Satria Budi Dharma Setia, PT Elokarsa Utama, and PT Sawita Interperkasa. And respect to all guests from Faculty of Mathematics and Natural Science, Chairman of the Yayasan Satria Budi Dharma Setia, presenters, and participants from Indonesia and overseas countries. Thank you for your attendances. It is our honor to have all of you here.
I'm honored to introduce our Dean of the Graduate School, Professor Doctor in Medicine, Dr. Setiawan. He has been a pillar of support of our academic community, and we are grateful to have him here to share a few words. Please join me in welcoming Professor Doctor in Medicine, Dr. Setiawan, to the stage. For your welcoming speech, the floor is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Our distinguished guests, ladies and gentlemen, in particular certainly distinguished speakers from abroad and also from Indonesia. On behalf of the Graduate School of Universitas Pajajaran as well as the Faculty of Mathematics and Natural Sciences, I would like to express our sincere gratitude to all of you for attending and also actively participating the international conference in biodiversity and biotechnology. So this virtual international conference is held as a collaboration between Department of Biology, in particular study program of biology for master, as well as the study program of master and PhD in biotechnology, Universitas Pajajaran. I think this conference is very important as a great opportunity for us to share the latest studies in the fields of food security and the sustainable aspect, and in particular also for students to share their current research in that field. And we are very happy to have 8 prominent speakers from Indonesia and also from abroad, namely from India, Malaysia, Japan, Turkey, and also Thailand. I certainly hope that this conference will give us opportunity to make valuable contact with international community, in particular in the field of biodiversity and also biotechnology. And eventually, we expect that we can have a good and productive internal and also external networking opportunities. If you look at the conference topic, it covers several issues, namely about biodiversity and ethnobiology, plant science for food security, animal science for food security, microbiology and food security, biotechnology for food production, agriculture, aquaculture and fisheries for food production, and last but not least, also about sustainability science and development. I would like to take the opportunity in my speech to a little bit introduce our schools. Our graduate school has several study programs related to the sustainability, environment, in addition to biotechnology. So we certainly welcome you all to get more insights about our schools, about our university, and we look forward certainly to our continuing collaborations. On this welcome remarks, I would like to thanks to all of you, in particular to several keynote speakers. The first is Professor Jyoti Prakastamang from the Department of Microbiology School of Life, Sikkim University in India. And also Dr. Namrata Tamang from the same department, namely from Department of Microbiology School of Life, Sikkim University, India. And also Professor Hitoshi Iwahasi from Gifu University. And then Dr. Wan Abdul Al-Qadir Imad Wan Mukhtar from Faculty of Science, University Malaya, Malaysia. Dr. Abdullah Bilal Öztürk from Yildiz Technical University, Turkey. And speakers from UNPAD, our Professor Johan Iskandar from Department of Biology, Faculty of Mathematics and Natural Science. And then Professor Ibnu Dwi Buwono from Faculty of Fishery and Marine Science. And last but not least, Professor Suseno Amin from Faculty of Agriculture, Universitas Pajajaran. Once again, I do hope that you can have a nice and fruitful discussion and insightful talk during our two-day virtual conference. And certainly, I look forward to continuing collaboration among us. Herewith, I would like, by reciting Bismillah, to declare the opening of this virtual international conference. Thank you very much. I wish you enjoy your day and also your scientific activity during this conference. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Doctor in Medicine, Dr. Setiawan, for those inspiring words for our conference. Moving forward, we are privileged to have a presentation from our esteemed sponsor, Yayasan Satria Budi Dharma Setia. Their support has been instrumental in making this event possible. Please join me in welcoming their representative to share a few words with us. For Dr. Vincentius Simeon Weo Budio, the time is yours.
Dr. Vincentius, are you ready with us? Oh, you can share screen with your own Zoom. Uh, okay, thank you for the information. We will make you as a co-host. Thank you. Please wait for a minute. Okay, it's done. Thank you. Thank you for that wonderful presentation and for your generous support of our conference. We are really grateful to have partners like Yayasan Setia Budi Dharma Setia with us. The next item of our agenda is the keynote speech, which will be moderated by Mr. Febri Doni, PhD. His expertises are in the plant microbiome interactions, phytomicrobiome, symbiosis, the system of rice intensification, and agroecology. Please join me in welcoming Mr. Febri Doni, PhD, to the stage. The time is yours.
For you.
And do you have a color mic so that it is convenient?
Punya ini enggak mic yang
Color
kecil
mic,
gitu.
otherwise I have to hold it.
Ya, Prof-nya nggak bisa pegang gitu yang lama.
Did you check my? Okay, this is tamam. Come here. Oh, okay. Hello. Hello. Sorry. Hello. It's okay? Started or no? Not yet. What time? I need a water also. What? You will read my CV?
Yeah, I'm the one who
Okay. Yeah, you can.
actually I got your CV from you're very famous, so
Okay.
yeah, even you
But
have
you
a
can
good
just read it.
I can read your CV from here.
Oh, it is coming.
Tes. Saya yang ini mulai. Okay, assalamualaikum.
Start up?
Yeah, we started. We'll start it. Assalamualaikum warahmatullahi wabarakatuh. A very good morning to everyone who is joining us offline and online today via Zooms. My name is Febri Doni. I'm gonna be moderating this keynote session today on fermented food. Well, we know that fermented foods constitute a significant component of human nutrition, dietary supply, and calories intake in different parts of the world, including Indonesia, where we have a lot of traditional fermented food, for example, like tempeh. Today, our keynote speaker will be discussing about how modern technology such as multi-omics approaches can be employed to enhance the utilization of traditional fermented foods, especially in South Asia. Well, I would like to introduce to you all our keynote speaker today is Professor Jyoti Prakastamang. He is a professor in the Department of Microbiology, Sikkim University, India. He is a pioneer food microbiologist working on ethnic fermented foods and beverages of the Himalayan regions of India, including Northeast India, Nepal, Bhutan, Myanmar for more than 36 years. He has conducted pioneering studies towards understanding microbial compositions and delineating health benefit of fermented food and beverages. Prof. Tamang did his master degree in botany with microbiology from North Bengal University, where he won a gold medal. He got a PhD also from North Bengal University, and he had a postdoctoral fellowship with the National Food Research Institute, Tsukuba, Japan. And also he did another postdoc in Institute of Hygiene and Toxicology, Karlsruhe, Germany. Prof. Tamang won many awards, including United Nations University Women Association Awards in Tokyo in 1996. And he has a vast experience working on this area of the fermented food. especially in South Asia, where currently has more than 50 H index in both Scopus and also Google Scholar. So we're indeed extremely honored to have Prof. Tamang today with us in Bandung. So before we proceed to the keynote speak, I would like to remind the audience that if you want to ask a question, you can actually drop your question in the comment in the chat box. So Prof. Tamang will be given 30 minutes for presentation and 10 minutes for the follow-up Q&A. Prof, are you ready? Okay, Prof, the platform is yours.
Namaskar. First of all, I'm very grateful to the Dean, Professor Tida, Professor Savitri, Dr. Gilang, and other friends. I remember, but I do not remember the names. We have been knowing for last more than 12 years. And for invitations, me to Bandung. Is it okay? Can I continue? Just here. I'm not audible. Now it's okay? Audible? Am I audible? Am I audible? Audible? Okay. Should I repeat again? Okay. Namaskar. First of all, I'm very grateful to the Dean of the Graduate Schools of Padazar University, Professor, then my old friend, Professor Tida, Professor Savitri, then Dr. Gilang, then others. I remember them, but I'm sorry I could not remember their names. And we have been knowing for last 12 years. And for invitation me to Bandung. And this is my second visit. I visited in 2013 in Bandung. Well, today I'll be delivering the lectures on the metagenomics and multiomics approaches in some Asian fermented foods, because now there's the latest techniques are the multiomics approaches to understand the complex microbial structures and the traditional knowledge, what I term is the ethnomicrobiology of the fermented foods. Next slide, please. Oh, okay, I'll do that. This one? The very important is why we eat foods. We eat foods for nutrition and healthcare, for the growth and the development, energy repair, and the maintenance of the body cells. Due to diet-related diseases such as obesity, cardiovascular diseases, diabetics, etc., the concept of the food as medicine is increasing among the consumers for the prevention and treatment of illness. Right choice of the food as medicine can impart the health benefits, strengthen the gut microbiota, and helps in combating diseases. Some of the important factors which influence the disease risks, such as dietary habits. Now we change our dietary habits, what we have adopted from our parents, grandparents, and all. Feeding frequencies, now we have changed all over the world. We do not have a fixed time for the breakfast. Sometimes we we skip lunch. Sometimes we skip breakfast. That will also affect and all. Then quantity and the quality, we have to give the more emphasis on the quantity rather than the quality of the food. That the type of culinary. What is fermented foods? The fermented foods are defined as the foods made through the desired microbial growth and the enzymatic conversions of the food consumptions. But I defined it as a knowledge of the food fermentation of locally available raw materials of the plant or animal sources, either naturally or spontaneously or by adding the starter cultures containing functional microorganisms which modify the substrate biochemically, organically, leptocally into the edible products that are culturally and socially accepted and all. Now, okay, so you can take it out. Am I audible? Am I audible? So there are more than 5,000 varieties of major and minor fermented foods in the world, which are eaten as a staple, curry, side dish, and etc. The ancient people knew how to domesticate the essential and the functional microorganisms from the environment for the production of the fermented foods. We authenticated scientifically domestications of the biofunctional microorganisms. Now, if you see this map, it's very interesting and all. If you see the black color is the Asian countries. There are 42 Asian countries, and mostly in the Western, starting from the Russia, then so many other the countries, Middle East and all. It is mostly dominated by the fermented milk products and followed by the fermented cereals and the fermented meat. But if you go to the eastern part, is mostly the circled by the red, is mostly dominated by the fermented soya beans and followed by the fermented vegetables and the dried amylolytic starters like ragi in Indonesia, then the many fermented fish and the meat products. And if you see in the southern part of India, the blue circle is a very unique. They do not ferment separately, but they mix it, rice and the legumes. That is what is idli and dosa. Now, these are the Asian ethnic foods, different other foods. And if you see the Indian, the gastronomy, the food distributions, that you see that the red circle in the east is mostly the fermented soybeans. Fermented soybeans are eaten in the eastern part, northeastern part of India, followed the fermented vegetables. In the western part, it is north and western part, it is mostly the fermented milk products, fermented cereal products, and so on. And as in the south, I told you it is the fermented rice and the legumes. The concept of the ethnomicrobiology is a very commentable indigenous knowledge of the ethnic people for production of culturally and socially accepted fermented foods. I coined this term, ethnomicrobiology. Why? Because the ancient people learned how to domesticate the essential and functional microorganisms from the environment for the production of edible fermented foods by using their ethnomicrobiological knowledge. One of the very prominent examples is the tempeh fermentations. The Indonesian people knew how to domesticate the essential microorganisms. That is what we have identified as a rhizopus, and it was not known to them. So this is how we have to do, because the fermented foods are the biological hubs of many functional life microorganisms. Now these are the techniques to compile the microbial the profiles, and traditionally, conventionally, it is called the cultural method. That means you isolate the the samples, you get the samples, you make a homogenizations, then you make a dilutions, then you plate on the culture plate, and whatever organisms grow on that plate, you just pick up the colony and identify it phenotypically, then morphologically, and you identify. But you miss many microorganisms which cannot grow in that culture media at all. So there are unculturable microorganisms also. Now the scientists have discovered another technique, what is called the culture-independent methods. There you don't have to get the sample, just extract the DNA or RNA, if it is a metagenomics DNA, if it is a transcriptase RNA, from the samples directly, and you do the sequencing. There are so many sequencings and all. So there are two very important sequencing, the microbial community. One is the high throughput sequencing, another is the shotgun sequencing. Now the high throughput sequencing is that I told you from the samples you extract the DNA, then but it targets only one gene. Particularly, suppose if you are concentrating on the bacteria 16S, it will give the only identification of the bacteria, lactic acid bacteria, non-lactic acid bacteria, all type of the bacteria, but not the fungi, not the yeast, not the archaea, not the virus. Again, you have to do it. But the latest technique, which is very, very common now, and the most reliable and most authenticate and the very powerful tool is the shotgun sequencing, is to profile the entire microorganisms. Here you extract the DNA and you run the sequence, and that sequence will give the entire microorganism or microbial community, whatever present in that food or in the plant or in the soil or clinical sample, any samples, you get a virus, you get a bacteria, different types of the bacteria. These are called the domains. Then the yeast, the fungi, then the archaea, then the virus also. So application of bioinformatics is very important. When you get the, I think this is a pointer. This is a pointer. No? Oh, this is okay. So once you get it, then in the third assembly, you get a just like your ECG, heartbeat, the ECG, the graph, which is you cannot read it. So to make it the readable form, to make it in the taxonomical form, you need a tool of the bioinformatics, which is very, very important. With the help of the bioinformatics, then only you can get the entire, the readable form of the taxonomic, what we call the meta-taxonomic. So then another technique in the metagenome is the metagenome assembled genome. I think you have heard the whole genome sequencing. Whole genome sequence is when you identify any bacterium or any yeast, any fungus. So you you extract the DNA, then you make a map, and where you can get the different type of the gene, the location of the gene, their properties and all of that particular. But now, with the help of the metagenome assembled genomes, you have a metagenomes. Automatically, it will be screened, and the high quality metagenomes will come with there are these times where you can you can just decode the different biomarker genes for the health and the functional attributes. Now, transcriptome is also very important, one of the very important omics. The transcriptome means when you extract the RNA, because there are many legume-based products like soybean fermented legumes, like in tempeh and all, where is a protein synthesis is very important and where the RNA plays a very important role. So the gene expressions can be done by these transcriptomes, and it can also give the metataxonomic of that. Now the metabolomics is very, very important. Metabolomics is a study of the small metabolites. What are metabolites? These are the end products of the metabolism. Nowadays, we are not concerned how much protein we take it, how much carbohydrate we have to take it, how much fat we have to take it and all. That concept has gone. Nowadays, every consumer is concerned how much bioactive compounds we have to take it. What are the different vitamins we have to take it? What are the different immunomodulators which can save our immunities, increase, strengthen stimulate our immunity? These are the things, biopeptides. So these are mostly present in the metabolites. There are two types of metabolites. One is the secondary metabolites, also known as the targeted metabolites. These are amino acid, fatty acid, and all which are used for the growth, cell development and all. But there are other metabolites which are also called the primary metabolites or the untargeted metabolites. Nowadays, the importance of this untargeted metabolites is very increasing because they are very small biomolecules. They can go to any part of the body, and this includes your immunomodulators, especially during the COVID time. Everybody knew that we have to increase our immunity. But what is that? There's immunomodulators, which are present in some plants and also reported in some fermented foods. So we believe that the consumption of fermented foods is very important to increase these immunomodulators. I will show some of the slides. Now there is an integrated metagenomics, metabolomics approaches. I'm not going into details of that. Now the CRISPR is also very important. I think it might be in your syllabus, which is called the clusters, regularly interspaced, short, palindromic, repeat CRISPRs associated Cas proteins, which are constitute the adaptive immune system in the several bacteria and the archaea. And now the proteomics, the metabolomics is mostly for the targeted, untargeted metabolites, and the proteomics is mostly for the protein-related and all. And this can be correlated with the transcriptome and the proteomics. Now there are 10 different types of the global fermented foods, fermented vegetables, legumes, cereal, milk, fish, and so on. Then now I'll go to the fermented soybeans. Soybean is believed to be first introduced in the Mongolia. Now it is Inner Mongolia in China, but initially it is in Mongolia. Wild varieties of soybeans were first introduced to China during Zhou dynasties around 700 BC, and the wild varieties were then first domesticated in the eastern part of northern China, which was considered as a primary gene pool, and it came to the Union, that's the southern part of China. Then from there, it diversified, one from to the Korean Peninsula, the Korea, Japan, another went to the northeast India, the Myanmar, the northeast India, the Nepal, Bhutan. Then another went to south, went to the Thailand, then Malaysia, Indonesia, and others. This is how this has been authenticated, documented. Now there are three types of the fermented soybean foods. First is soybeans fermented by the filamentous fungi, yeast, and the lactic acid bacteria. Mostly example is the soya sauce, soyu, tamari, and miso, etc. And another is soybeans fermented with bacteria, mainly the Bacillus species. This consists of the whole beans that become sticky, non-salty, and the highly slightly ammoniacal, and restricted to the few Asian countries, so-called the Kinema, NATO, Tuano Triangle. I'll explain you later on. Then third is the soybeans fermented by the mold, mostly by rhizophores, example the tempeh of Indonesia. Now this is very important. The Bacillus fermented, sticky fermented foods are confined only to this, the hypothetical triangle, which I coined as a Kinema, NATO, Tuano Triangle in 2010. If you see in the eastern part, this part is right from the Nepal, it is called Kinema, and Bhutan and Nepal in India, in the northeast India. Then in Myanmar, it is Pibok, and Thailand, northern Thailand, it is Tuanao, and in Cambodia, it is Siang, and in southern part of China, it is Daochi, and Korea, it is Chungkokjang, and Japan, Nato. So if you draw the triangle, is a triangle, imaginary triangle, what we call Kinema, NATO, Tuano Triangle. The interesting part is that, though the soybeans, both fermented and non-fermented soybeans are consumed in mostly the Southeast Asia, but the sticky and the ammoniacal flavor fermented soybean foods are confined only these countries. You don't find this sticky fermented food in Philippines, nor in Vietnam, nor in Indonesia, Malaysia, and all. Now these are the different types of the soybeans, and in India, the northeast, there are varieties of the fermented soybean foods with a different name, Kinema, Hawaiijar, and so on. And in China, it is called Daochi, Tuanao, I told you, and in Japan, it is Nato. Now I'll just give an example of how one of the fermented foods, Kinema, is very popular and one of the oldest fermented soybean foods of the eastern Himalayas. These are prepared traditionally without any starters. So what we did is we collected these samples from the different parts of it, Nepal, then India, then the Bhutan. Then we did both hydropod sequencing and the shotgun sequencing and all. Then what we found in the hydropod sequencing, we detected 277 bacterial and 80 fungal species. And though the Bacillus subtilis is the dominant, with the other species, 27 species. Then in recently, we did the shotgun sequencing, the latest technique to profile the microbial community. We detected 1,560 species or domains, where 1,451 bacterial species, 42 viral species, 56 eukaryote species. It includes the yeast and the fungi, then 11 archaeal. Then very interesting is 280 species of bacillus were detected. Now this is very, very, very important. How we have authenticated the traditional knowledge. By the cultural method in 1990, we identified only 3 species. That is the Bacillus subtilis as the dominant species. In 2019, with the application of the high throughput amplicon sequence, we identified the 27 species of the bacillus from the same kinema samples. I mean, not samples, but the same kinema traditionally prepared by the Himalayan people. Then in 2022, by the application of the shotgun metagenomic sequence, we found the 280 species of bacillus. And this is the Bacillus subtilis, which is very highly functional, health promoting, the very high, the health promoting, the functional beneficial bacterium and all. This is what I want to say, that the advent of the metagenomics, shotgun metagenomics, the tool is very, very powerful. This kinema has been consuming for more than 5,000 years. That people did not know what are these bacillus, what are the different species and all. But now with the help of these techniques, we could identify that this is a huge microbial diversity present in the fermented foods. Only the thing is that we do not have that technique. Once we have a technique, maybe after 5 years, in 2029 or 2030, this may be the advanced technique that will become or superset this shotgun metagenomics, which will be obsolete, and maybe in that way, maybe more than 1,000 or maybe 2,000 species of bacillus may be detected. Now we did the metabolomics also. We found 28 compounds, different type of the untargeted metabolites in kinema. Then these are the different type of the metabolites. We found that the potential immunomodulators, different types of immunomodulators, different type of the bioactive compounds, and different sapponins, then the vitamins B7, B3, B6, and so on. So they are very rich. And the flavor also, we did the flavor analysis also, GCMS. And we found that the very dominant flavor is the umami flavor, the glutamic acid. The flavor is being generated when the protein is broken down to the amino acid. The flavor is that flavor is called the Japanese term, the umami flavor. It is nothing but it is mostly the glutamic acid. Then we did the MAC also, metagenomic assembled genes, where we found the different type of the function, different types of genes. And these genes are the biomarkers of the genes for the bioactive peptide productions, the vitamin biosynthesis, fibrinolytic, the secondary metabolites, and all. These genes are present. So this is very, very important, the data. And we also, because it is a sticky, we also analyze the polyglutamic acid, which is called the PGA, and it has got the very high the PGA productions, and has got the very, it regulates the blood pressure, helps in the calcium absorption in the intestines. And we did the therapeutic and the anti-thrombotic properties also. We found that some of these bacillus species produce the fibrinolytic enzymes, which are very important for the heart disease, anti-thrombotic activities and all. Then some of them have also the probiotic properties and all. Then we did the similar type of the foods in the Arunachal Pradesh. We did the same metagenomics and metabolomics, and we did the MAC. I'm not going into details of this. This is how we have done. Now in 2019, we visited Korea. Then we collected Chungkokjang with the help of some of my friends, and we brought to my laboratory in Sikkim University. We did the metagenomic shotgun, and this is the first paper on the shotgun metagenomics, Chungkokjang, the similar fermented soybean food of that natto Thwanot, kinema natto Thwanot triangle. Then in 2020, I visited Myanmar, and there the fermented soybean is called the pibok. Then we collected the samples and we brought it to my laboratory. We did the shotgun sequencing. I think this is the first paper on pibok from Myanmar. Then recently, just 2022, last year, we visited Cambodia and we collected the shang, and we also did the shotgun metagenomics and the MAC, and we published in the Food Biosciences recently, up to 3 days ago. And this is the first paper on the shang of Cambodia. Then we compared with the different sequences of all that. The Bacillus subtilis is very dominant and different types of species and all. Then there are more fermented soybean products like soy of Japan, tempeh of Indonesia, then jjimjang of Korea. Now, and sofu, the tofu is non-fermented. If you ferment, it is called the sofu, and then the miso and all. Now the tempeh. This is, we have written a review published in the current opinion in Food Science last year. This is ethnomicrobes of tempeh. Indonesians have been historically domesticating the golden mold, the Rhizopus oryzae, to produce tempeh, a fermented soybean food in, fermented soybean food in Central or East Java in Indonesia prior to 1800. By using their indigenous knowledge of the ethnomicrobiology. This is very, very remarkable. Tempeh has several functional properties, health benefits, therapeutic uses. Some metabolites have also been detected, analyzed. Metagenome analysis of tempeh has also been done. Metagenome assembled genomes from tempeh has also been done. This has been done, but I could not find the transcriptomics and the proteomics. Maybe some of the researchers are working on this. Now we have an ethnomicrobiology of the fermented milk products. Consumption of milk and milk products can be traced back to 900 BC. The lactose, which is sugar in milk, intolerance. Many people are lactose intolerant because they cannot digest the lactose, which is sugar present in milk among the Mongolian races. But this can be supplemented by the fermented milk products. The fermented milk products which contain low or no lactose. So drinking fermented milk products reduce the symptoms related to lactose intolerance milk. And these are the different type of the fermented milk products in India. And this is like dahi of India, then dadit of Indonesia. Probably the term might have come from India, dahi. It is called dadit, but dadit is prepared from the buffalo milk in Indonesia. And there's a tarak of Mongolia, kumish of Kazakhstan, arak of Mongolia, and so on. So we also did the bacterial community of this. I'm not going into details of this. Then we did the probiotics, then the different type of the probiotics from the bacteria also, probiotics from the yeast also. These are from the bacteria. Then from the yeast also we have done, because some yeast also are the probiotic, the potential probiotic producers. And we did, this is a very interesting paper, we did the whole genome sequence of the potent probiotic. Recently we identified is a Levylactobacillus brevis from one of the indigenous product called churpi, fermented milk product. So it has got a very high GABA producing, the whole genome sequencing, and this is the picture of the Levylactobacillus brevis, lactic acid bacterium. And it has, we have annotated the whole, the biomarker genes and all. Now, at present, we are developing it as a starters. I am collaborating with the leading, the milk dairy company in India, what is called the mother dairy. I have already sent the cultures. I have already had a meeting with the chairman and the scientists of this group. And hopefully that we will be develop a probiotic Indian the product. And we have the lal dahi. Lal dahi is a mishti dahi, is a sweetened dahi. It's a very important. It is in the eastern part of India. And though the cultural, the identifications has been done, but nobody has done the metagenomics. We collected these samples and we did that, the whole, then so many the microbial community structures were detected in the lal dahi. Then, of course, we did the MAC also, and we found many functional biogenes, biomarker genes for the health promoting benefits and all. Then there are fermented vegetables, which is called the gundruk in the eastern Himalayas, and in the similar product in Korea is called the kimchi, saukai in China, sauerkraut in Germany. So this is the traditional prepare of gundruk. And there also we found the Lemilactobacillus, Lemicilactobacillus fermentum, lactic Cassibacillus cassi, and all. So there are many Indian fermented cereals also, the naan, jalebi, roti, and all. And then jalebi is one of the very important. I think it is also available in Indonesia, especially in the Indian shops. This is a batter. We collected this batter, and this has not been done. And recently, we did a shotgun sequencing, and we found the paradigm seed. So many huge microbial diversities have been detected in this jalebi with so many biomarker genes for the health benefits and all. So then we compared the genomes, metagenomes of the jalebi and the sauerkraut. Sauerkraut is a fermented sour bread of America and Europe. And this is what I called you the mixtures of the cereals and the legumes, that the idli and dosa. And this fermented, initially it was believed that the leuconostoc messenger varieties was the initial the bacterium for the fermentations in 1965. But recently, with the advent of the high throughput sequence, it is said that the Vishela is the main initiator at 9 hours, then Enterococcus, then Streptococcus. This is how the paradigm shift from the conventional to non-conventional. The approaches are very important. Now there are the alcoholic fermented beverages. Well, winemaking, brewing, malting of the beer is not a history, not only India, in the Southeast Asia. Though now everybody, everywhere there is a beer, winemaking and all. Actually, the Asians, they make the beverages, the alcoholic beverages, by the starter cultures. There are different types of the starter cultures with the vernacular names in India, and we did the high throughput sequencing, and we found the different types of yeast present in the starter cultures. Now, I'm not going into details. The similar type of the starters in Indonesia, it is called the ragi. In Korea, it is called the nuruk. And main in Vietnam, yung and Q of China, lokpang in Thailand. So if you see the diversity of these starter cultures, like the ragi in Indonesia, the different countries in Southeast Asia have the starter cultures, which contains the filament molds, which contains the filament molds, amylolytic and the alcohol-producing yeast, and the probiotic lactic acid bacteria. So these are all our publications on these amylolytic starters and all. So this is one very important. This is the International Year of Millet. So millet has been consuming in the Himalayas for more than 5,000 years. We found the vitamin B12 is produced by the fermenting microorganisms. It's very rich in amino acid, very high calorie, mostly given for the postnatal woman and the ailing persons, and the alcohol content is less than beer, 4.8, and there is a availability of the minerals. Now, another important is the fermented palm drink. So what we call in India, in Africa, and other countries is called the toddy. So we did both hydrofruit and the shotgun also. We found the huge microorganisms on this, and we compared the metagenomes of the toddy and African fermented palm and the pulque of Mexico also. Now this is, I think this is my last slide, and this is very important. In 2013, I visited one village escorted by Dr. Gilang and Professor Tida, and I wanted to see the tape fermentations and all. So how they do is, traditionally, tape is fermented by the cassava. Cassava is boiled and cooled, and the starter cultures, what is called the ragi. Now you get the commercial ragi, and it is packed in the banana leaves and just loosely tied and keep it for 48 to 24 hours to 48 hours depending on the seasons. And you find the next day it is full of the mycelium and it is so sweet. So after that, traditionally, it is being made airtight and make it the glycolysis cycle, then the alcoholic fermentations, so you have a tape. But nowadays that has been quenched. So they are not interested in the alcoholic beverages. Now it is only up to the tape, the saccharification. What is scientific reasons is the cassava is very rich in the starch, and they have got the toxins. When by boiling, is detoxicated, and after that the ragi is being sprinkled or the mixed, and ragi has the filament molds, the yeast and the bacteria, mostly the amylase producing the fungi and the yeast. They break down the starch into the glucose. So by loosely, then if you see the moisture and if you test this tape, the end product, tape, it is so sweet, just like a sugar. Then I found that in that village, I do not remember that name of that village, I think it is 1.5 or 1 hour away from Bandung. And I found the big, the truck, air-conditioned truck. And I said to Gilang, why are they? Are they tourists? No, sir, they are not tourists. They are coming from the bakeries from Bandung, Jakarta to take this. So they sell it. The people are sustainable. They are economically dependent on this, on the bioresources. So they sell, actually they are selling the natural sugar. So this tape is being made a paste and it is mixed in the bakeries. So whatever the bakeries in Bandung or Jakarta you get there, I believe that they are not using the sugar, they are using the natural, the glucose, which is very, very important and all. And this is one of the best pictures, and this is one of the best examples of the ethnomicrobiological knowledge of the Indonesian. Wherever I go in any part of the world, I am now advocating this. I request this university, the dean, to take up this and take it to your president, to your ministries, and make it a highlight of this. And this has to be grown, and so that the people will economically depend on that. There are some clinical tested popular health promoting, only few, not all. So which has got the anti-diabetic, antioxidant, anti-carcinogenic, and etc. That is the Yakult product, yogurt, which is marketed by the Japanese Yakult and Akifit companies in Europe, and kimchi of Korea, nata de coca of the Philippines, then kefir of Russia, and kombucha of China, which are available in the international market. So health benefits of the fermented foods have got several health promoting foods. They protect from the hypertension, nutrient supply synthesis, then lowering blood sugar, they prevent the cancer, and so on. Now in the conclusion, traditional and historically, the Asian food habits do not require additional medicine or supplementary drugs. If you see your parents and your parents' parents, and if you are lucky at your home, if you have a grand or great-grandparents who are more than 80 or around 90 or so, more than 75, even still they do not take the medicines because they believe on the food and all. So the present-day consumers are more concerned on their health and the food preferences, which cure the illness, maintain the blood sugar, control the blood pressure with the slim, and look younger. Always people are concerned of that, because I told you, people are not concerned on the protein, carbohydrate, this and that, but bioactive compounds. Some Asian fermented foods have been scientifically studied and proved to have several health-promoting benefits to the consumers. However, health claims of Asian fermented foods, except few Japanese and the Korean, based on the human clinical trials, are yet to be conducted to authenticate their therapeutic values. When you claim in the commercial market, when you industrialize, or in any fermented, traditional fermented food in the market, you should have a data on the animal studies. Animal studies, yes, some of the many fermented foods have the animal studies, but the human clinical trial is very important because every human clinical trial of every country is controlled by the regulatory authority of that the government. Every government has its own policy. I do not know the Indonesian government has constituted the clinical trial guidelines for the fermented foods or not, unless and until you cannot claim. And if you cannot claim, you cannot market it. Please remember, those who are doing, they are all false stack. It has to be done clinically, tested that this food cures the cancer, this food cure the blood pressure, this fermented food cure the diabetes. It has to be done clinically, human studies. Application of metagenomics, multiomics integrated approaches, including the MAC, may validate the traditional fermented food as medicine. So in our university, it's a national university, Singham University, and we have in 2020, we have revised our syllabus of the national education policy, multidisciplinary approaches in the 4 semesters in the PG, in the graduate. We have the 2 papers, the food microbiology, where we have included the 4 units, the 4 credits, the traditional food fermentation. Students should know about the traditional food fermentation, microbiology of the fermented foods, and the health benefits of fermented foods, and the gut microbiota. Today I am not talking about the gut microbiota because it is another subject and it takes a lot of time because I have a limited time. Then another paper is the functional and nutraceutical food, the nutraceutical food, prebiotics, probiotics, and allied biotics, the bioactive compounds and the untargeted metabolites and fermented foods. That the foodomics is very important. If you have a nutrition, now the foodomics in the genomic level has to be studied and all. So these are all my publications, and some of these are maybe in your library, and you can see in the Google, published from the Thailand Francis, Sprinkle Nature and all. And this is my team, my old and present PhD students. I've already produced 16, and another 10 are working on that. They are placed in the different university research institute. This is my current PhD students working in my lab. Dhanyavad. Namaskar.
Thank you. Thank you very much, Prof. Tamang, for your insightful presentation. And indeed, we have learned a lot from your presentations. Well, Prof. Tamang has presented many interesting insights on fermented food in Asia, as well as he already explained to us, including transcriptomic and metabolomic analysis, using the machine learning tools to characterize microbes that are present in the starter cultures and fermented food to contribute to their health benefits. Prof. Tamang also already presented and very well on the various fermented food in Asia, in Asia, and also we can see the maps that Prof. Tamang has shown to us that indeed there are a lot of fermented food in our region, Asia. So thank you very much, Prof. Tamang. So we're moving on to the Q&A sessions. So we have a question from, yeah.
Hello.
Yeah.
The participant who want to.
Yes.
The following is my question. How is the impact of long read and short read sequences of food research? Oh, this is very good questions. Because see, long read can give you the more depth in informations. What is called the in-depth bioinformatics, because the sequences has got the all informations and all. So that it will be, because we detect only more than 1%, but there are more than, even below 1% also, there are very huge diversity of the microorganisms. And I think that, and some of the bioactive compounds by the predictive functionality also. Can you show the other question? No, up. Okay, this I answered. Then I don't know, it is written in Indonesian, so I cannot understand. Can you translate it?
I think we can move on to the last question in English. Then I will translate.
Hello.
Yeah.
I would like to ask you about the functionality value of the fermented product. Currently, there are many modifications to fermented foods product, such as yogurt with the addition of the chocolate or some with the edit and all the functional value of the. Okay, now see the first of you have to know the intact the food value or the functionality of the traditional fermented foods present in that fermented foods which are traditionally prepared. After knowing the whole background of that, once you have the metabolites, the proteomics, then all type of the amino acid, whatever the bioactive compounds you have, different targeted, untargeted metabolites, then you can modify that if you go for the commercial. So in the yogurt, as I told you, yogurt has been clinically done, human clinical trials have been done, several health claims have been done. Now they can, that is what we call the fortify. They can fortify or diversify the recipes. Some of them are mixed with the different fruits, some of them are mixed with the chocolate, some of them mixed with the thing also. That can be done, that's not a problem. Thank you for your presentation, Prof. I am Shiti, student of PhD Gilang. I have tried to use the PICUS to, there's a pipeline to predict of the 16 years, and that's a high throughput sequencing analysis, connect with the metabolism, whether it is strong enough to predict. The production of an enzyme in the process. What high throughput method is more recommended to see the potential microorganisms to produce the enzymes and metabolites? Now, you see, you can predict with the PICUS, and there are different new versions of the pipelines, and all these softwares are free of cost, but you should have a knowledge in and the practice in the bioinformatics and all. And this you can do only from the metagenomes or the reads which you have that also. But if you want to correlate with the real-time research, and that is the metabolomics, that GCMS, LCMS combinations, you should run that instrument and you have the data from that. Now with this data and with your metagenome data, you can correlate. But in the high throughput sequence, it is not so possible, and it's a possible but not so reliable. So you have to go for the shotgun metagenomes. If you have a shotgun metagenomes, you can use the metataxonomics also, identifications of the microbial diversity, profiling also. Then at the same time, you can correlate the different type of the metabolites. If you do the metabolomics, if you do the proteomics, also protein. Next. Thank you.
Okay, Prof. I think I have.
Last. Okay.
Yeah, I think I have one question for you.
Okay.
I was wondering how transcriptomics can be employed in studying microbes in fermented foods. Is it we are studying the genes from the microbes? What we should do?
No, I didn't understand
yeah.
the question.
I was wondering how transcriptomic
Transcriptomics.
yeah can be employed in the fermented foods study. You know what the genes that
that's
being
a good question. I have already showed in my slide the different applications of the omics. One of the important omics is the transcriptomics. The transcriptomics is mostly related to the gene expressions, the protein synthesis. And in the transcriptomics, you have to extract not DNA, RNA. You have to purify the RNA, you have to extract the RNA. And from RNA, what are the genes located, present there, and what are the role in the gene expression, mostly in the protein base and the protein synthesis. Yes, you can do that also. Then after getting the information of the entire gene expressions of that particular organisms, or you can do from the particular organism also, or you can do from the particular food also. Then to make it more, what are the different types of product? You have to do the proteomics. The proteomics, what are different types of the amino acid, what are different types of protein, and you can correlate, just like metagenomics and metabolomics, transcriptomics, and the proteomics. But these are the very rare papers. I think very few, very, very few, as I think, of course, we are working on that. And I'm interested to work some of the fermented foods of the Indonesian and the Asian, also using this transcriptomics and the proteomics.
Okay, Prof. Thank you very much. I think time is out now, so I think that's it, Prof. Thank you. Thanks again, Prof, for your very insightful presentation. Because time is out, and I'm gonna give back the platform to the uh, Aji and Rasi. Yeah. All right.
This is soundproof. You use this asbestos. Soundproof asbestos. There is a oh, number one. Now next. Dr. Number. Dr. Number. She will be. You have no?
Thank you for Mr. Febridoni for your outstanding moderation, and Prof. Jyoti Prakash Tamang for a great keynote speak. The next agenda is a keynote speech by Professor Johan Iskandar, which will be moderated by Dr. Dina Octavia. Dr. Dina Octavia has expertise in ecology, biodiversity and conservation, ecotourism, conservation and restoration, and natural resources management. Please join me in welcoming Dr. Dina Octavia to the stage. The time is yours.
Hello? Check. Check. Good morning, ladies and gentlemen. I'm Dina Octavia, and it is my pleasure to be the moderator for the keynote speech session 2 on the virtual international conference on biodiversity and biotechnology for food security and sustainability. In acknowledgement of the relationships among biodiversity, resilient ecosystem, and the conservation of indispensable genetic resources for Indonesian agriculture, our upcoming session is dedicated to exploring the ethnobiology for sustainable development in Indonesia, where ethnobiology provides pathways to enrich agricultural productivity, foster the growth of crop varieties, and confront food security challenges based on holistic, culturally sensitive, and sustainable approach. Ladies and gentlemen, without further ado, now I will invite our keynote speaker, Professor Johan Iskandar, MSc PhD, to come with us to the studio. Welcome, Professor Johan. Okay, ladies and gentlemen, first I will read a brief introduction of our speaker, Professor Johan Iskandar, MSc PhD. He did a bachelor degree at the Department of Biology, Faculty of Mathematics of Natural Sciences, Universitas Pajajaran. And in 1991, he graduated with Master of Sciences in Agriculture Technology, Chiang Mai University, Chiang Mai, Thailand. In 1988, he graduated with PhD in Ethnobiology, Environmental Anthropology, University of Kent, Canterbury, the United Kingdom. Now, he is the Professor Emeritus at the Department of Biology, Faculty of Mathematics and Natural Sciences, Universitas Padjadjaran. And he is also a senior researcher at the Center for Environment and Sustainability Science, Universitas Padjadjaran. His research focus on human ecology, ethnobiology, biodiversity, management of agroecosystem, ecology of landscape, and ornithology. Professor Johan has published a number of books and more than 100 international journals. Okay, ladies and gentlemen, before we start the presentation, let me tell you how this session will run. The first speaker will present the material for about 30 minutes, and after that, there will be 10 minutes for the question and answer. Well, ladies and gentlemen, now allow me to welcome Professor Johan, MSc PhD, to present the material entitled Ethnobiology for Sustainable Development in Indonesia. The time is yours.
Good morning, everybody. First of all, I'd like to thanks for Professor Setiawan, graduate Faculty of Padjadjaran University, and also Professor Sapitri, who have invited me as one of the keynote speaker in the virtual international conference. In this opportunity, I am going to talk about ethnobiology for sustainable development in Indonesia. Four aspects will be presented. First, Indonesia has high diversity and rich cultural diversity. The second, biocultural diversity, that is diversity of life, is expression biological, cultural, and linguistic that are interrelated in biocultural diversity. And third, ethnobiology and ethnobiological research based on a case study in Baduy, South Banten, Indonesia, and fourth, ethnobiology and sustainable development, and the last one is conclusion. Begin with Indonesian biodiversity. Indonesia is a large archipelagic country that has rich biodiversity. For instance, Indonesia, it was recorded 4% of flowering plant species in the world, and 20% of fish species in the world, 17% of bird species, 16% reptile, 12% of mammal. So Indonesia has been known as the second ranking for bird diversity in the world after Brazil. In addition, Indonesia also one of the most rich cultural diversity in terms of language. For instance, Indonesia has recorded more than 700 local language in Indonesia. So Indonesia has been known as the second high diversity in the world concerning the local language. So in 1990s, it was popular research concerning biocultural diversity. Cultural diversity is diversity of lives, of its expression of biological, cultural, and linguistic are interrelated in the development process in biocultural system. Or in other words, biocultural system, there is relationship between cultural diversity and biological diversity. So concerning diversity of culture and linguistic and biology, it has popular known concept of biocultural system, and also cultural system is a form of, as mentioned before, biodiversity is, and cultural diversity and linguistic diversity. So in Indonesia, it has been known several traditional agroforestry. For example, in West Java, we know about home garden, or we call pekarangan, and also has mixed garden, or kebun campuran, and also Sweden farming, or shifting cultivation. That is jointly shaped biological and cultural dynamic of the local people over time. So in 1990s, also have been popular research on local language, local knowledge. Local knowledge has some synonyms. For instance, indigenous indigenous knowledge, indigenous technical knowledge, ethnoecology, and traditional ecological knowledge. Traditional ecological knowledge can be defined as a body of knowledge, practice, beliefs, and evolving in dynamic system. And we have popular also the study on local language or local knowledge on biology. We are called ethnobiology, and ethnobiology can be divided into ethnobotany, ethnozoology, and ethnoecology. So if we call ethnobiology is a study relationship between people and environment. So we study about knowledge, practice, and knowledge of various communities, particularly traditional people in the villages. So last 5 years, we had research concerning ethnobiology with special topic ethnobiology research for supporting sustainable development program. We have some research about ethnobotany, ethnozoology, ethnoecology, and so on. So in this case, we usually conducting research in the rural people, and we interview local people to understand knowledge, practice, and belief of the local people concerning various aspects of biology. So the research we consider is very important, can be used as development of science, and some result for supporting sustainable development programs. So in ethnobiology or ethnobotany or ethnoecology, we want to understand about knowledge or corpus, and also a praxis and belief or cosmos of the local people. So in this case, usually we have analyzed for level of the local knowledge of the local people, including knowledge, practice, and belief of the local people. So this is one of example our research was conducted in Kanekes Village, Lewi Damar Subdistrict, Lebak District, Banten Province, Indonesia. So in this village reside traditional people we call Baduy community. Baduy community is popular as one of the traditional communities in in South Banten. They have some taboos and some regulation or some prohibition. For example, they have not been allowed to cultivate wet rice farming or sawah, but in return, conversely, they have a special Sweden farming. So according to Baduy people, the Sweden is considered as obligation of their religion. So every family of the Baduy people must conduct on Sweden cultivation or Sweden farming, but they are not allowed to cultivate wet rice or sawah farming. So based on local knowledge or traditional ecological knowledge, the Baduy has some landscape system in their area. For instance, Baduy has some landscape, traditional landscape. For instance, the forest. In local language it's called hutan or leweng, and forests can be divided into categories: the protected forest and non-protected forest. The protected forest is considered as a protected area and cannot be cultivated for Sweden farming, but non-forest area can be divided, can be cultivated to be Sweden farming. So every year, the Baduy community usually open the non-protected forest for cultivating upland rice mixed with other annual crops in the Sweden farming or in the shifting cultivation. So the protected forest is located in the upstream of the river. So that is important for traditional conservation area in the Baduy or in the surrounding areas. So the river can be considered as upper part and the middle and downstream is based on Baduy concept. Yeah, also in the valley, yeah, there is resettlement and circled by forest. It's a protected forest. Also, we call Baduy people usually call as village forest, and also is not allowed to cultivate, not allowed to cut the trees. This is as protected forest. In addition, also forest ground in next along the river is also protected forest. It's not allowed to cut or to open for Sweden cultivation. And in the settlement area, also there is some rice barn, or we call Leuit. It is a special place for storing production of rice in the Leuit or rice barn. So based on Baduy local knowledge or traditional ecology, hills, certain hill can be divided into 3 zones. The first zone in the valleys is particularly for settlement and circled by village forest, and also rice barn are placed in the settlement area. The settlement area usually is located near river or near water resources. And the second zone is for practicing Sweden cultivation. They usually open the certain secondary forests, and before rainy season, before beginning rainy season, they plant rice, and after harvesting rice, about 4-5 months, the Baduy people can move to another plot to open the secondary forest. But the third zones in the top of the hill, there is the forest, particularly the primary forest, is not allowed to open. There is a special for protected forest. Now, very interesting things in Sweden cultivation has been cultivated, has been grown by annual and perennial plants. Yeah. For instance, in Sweden cultivation has been recorded 69 species of edible plants, consisting of carbohydrate staple foods and non-carbohydrate staple food, also vegetable, spices, fruit, medicinal plant, and so on. So, in terms of rice varieties, also in Baduy has been recorded at least 89 rice land races or rice local varieties according to Baduy classification. The rice, the Sweden rice, can be divided into morphological aspect or ecological aspect, also gastronomic. For instance, in terms of morphology, some varieties can be grouped, can be classified as sticky rice and non-sticky rice. There is hairy and non-hairy rice, also white hulled rice, red hulled rice, black hulled rice, and now normal maturing. This is 4 or 5 months, and also early maturing. Early maturing before 5 months can be harvested the special varieties, and also rice can be considered as sacred rice and non-sacred rice. So the Sweden cultivation practice has conserved various local rice varieties. Based on Baduy, they are not allowed to use modern varieties, modern rice varieties. They usually cultivate local rice varieties. And based on our study on ethnobotany, it was recorded about 89 rice land races or rice local varieties. And also they are not allowed to use pesticide, synthetic chemical pesticide. They usually use biopesticide, such as botanical pesticide. And also they are not allowed to use a modern tool. For example, they're not allowed to use hoe to dig soil and so on. In other words, the Baduy community has a practice organic farming, and also popular term for modern agriculture at the moment. They have a practice on LESA, low external input, and sustainable agriculture, because in their cultivating of Sweden cultivation, they are not allowed to use various external input, such as rice varieties, modern rice varieties. They are not allowed to use synthetic pesticide, and also they are practice organic farming. So the varieties of crops, not only rice, but also some crops such as banana, Musa paradisiaca, cassava, Manihot esculenta, taro, Colocalia esculenta, also have various local varieties or land races. For instance, banana, based on our ethnobotany research, it has been recorded 45 varieties or land races of banana. They are called local term, such as pisang Ambon, pisang bakar, pisang mas, and various local varieties of banana. In addition, also cassava has been recorded 9 land races, various cassava, and taro or Colocalia esculenta also has several land races or local varieties. In terms of economy, Baduy has developed a special economic system. We are called as moral versus interest. In terms of moral, the rice varieties of Sweden farming are not allowed to trade it, not allowed to sold it. They are used especially for home consumption. So production of rice usually put in the rice barn, in the lid, but other crop productions such as durian, banana, jengkol, petai are allowed to sell. So they can obtain money, cash income from selling various non-crop, non-rice varieties. But rice is considered as sacred, and must be put or store in a special rice barn. So in the time recently or in recent time, the Baduy has high security of food security because they have a special stock of rice in the rice barn. Although at the moment has anomalic climate, yeah? Climate and weather. They are now also at the moment as dry season, long dry season. But for Baduy is because they cultivate rice and non-rice crop in the Sweden farming. So in terms of rice, maybe it's not good the harvest at the time, but some production of non-rice crop, for instance, durian, petai, jengkol, and so on, can be harvested good. The production is not seriously influenced by climate change at the moment. So based on Baduy economy, so as mentioned earlier, the rice can allow to sell or to sold or traded, but rice put in the special rice barn or and rice can be stored in rice barn for 10 or more than 40 years. Then it is still normal, can be consumed, but of course for the planting the next year and next after harvesting at that year, so they use new rice from the recent harvested, but the rice put in the rice barn still normal, can be consumed for family. In other words, the rice can be stored for more than 30 or 40 years in the rice barn. So we consider the Baduy has high food security. So in terms of food availability, so the various crop can be harvested in various traditional agroforestry, from Sweden farming, from village forest, from secondary forest. Secondary forest usually is planted by various fruit trees, and also shrub, wide shrub species, yeah. And also food access. Every family usually has Sweden farming, because according to their religion, every Baduy family has to own Sweden plot. So the food utilization, this is we consider has high equitability in terms of every family, and also stability over time, because they usually practice traditional agroforestry. So the production of agroforestry more stable than that of planting wet rice in wet rice farming system. So according to Baduy community, they have a special various medicinal plant which are usually used for treatings, for treating various illnesses or disorder health. According to ethnobotany study, it was recorded 10 various illnesses in Baduy. For example, there is intestinal worm, there is cough, there is diarrhea, and so on. And Baduy community has a special treatment for treating of the illnesses. They usually use various medicinal plants which are grown in the various ecosystem of the Baduy area. So this is one example. Various traditional, various local medicinal plants. Yeah. For instance, Blumea balsamifera, Curcuma longa, Clidendron serratum, Campharia angustifolia, Ginger officinale, Ginger riperum, are predominantly used for maternal postpartum recovery. So they have traditional to use various medicinal plants that are harvested from various local ecosystem in their village. So in terms of ritual, there is some ritual for planting rice because according to Baduy, rice is considered is a sacred plant, sacred crop, and according to Baduy, they reside in a special place. They consider the area as holy place, and this is not allowed to use pesticide because this is poison for the soil, and every stage of rice cultivating involve some ritual. Yeah, for instance, before cutting the secondary forest, they must conduct a special ritual offering for the ancestor. They ask or want to have permission from the God. They would like to success in rice farming, in practice rice farming in their area, and also as a special offering ancestor. About 4 species are usually used for offering ancestor, and before put rice in the rice barn, there are special also ritual. They call taking rice from the rice barn. It's a special ritual, and also about 4 species are usually used for that ritual, and the total of the plant that are usually used for various ritual in Baduy community recorded about 40 plant species, 28 families for conducting 9 main ritual. So Baduy, as mentioned earlier, they have practiced economic system, moral versus interest. So in terms of moral, the rice production are not allowed to sell or to trade it, but non-rice crop production, for instance, banana, durian, and so on, can be sold, and they can obtain cash income from selling the cash crop, the non-rice crops. So based on our study, we consider about the study of ethnobiology, ethnobotany. It has been important role for supporting sustainable development program. For instance, we can hybrid traditional ecological knowledge and Western modern knowledge for improving traditional agroforestry in the village area. For instance, the agroforestry system, traditional agroforestry system, has an important role for providing food and also for some benefit of ecosystem services, such as for production oxygen and carbon dioxide sequestration, for habitat of animal, and also for protecting soil from erosion and other benefit of ecosystem services. In terms of production, various products can be harvested from traditional agroforestry of Baduy. For instance, edible, vegetable, staple food, medicinal plant, and other function. So our conclusion, we have a special benefit of the study ethnobiology in Baduy case. The ethnobiology, ethnobotany has been important role for supporting development program, for supporting, for instance, the end of poverty, yeah, because their traditional agroforestry has sustainable, appropriate agricultural system, appropriate in terms of social economy, and also appropriate for ecology, local ecological system. So from the study, we conclude the ethnobiology research has been important in terms of development of science, and some research can be used for directly for supporting the sustainable development. Thank you. This is my presentation. Thank you very much.
Okay, thank you very much, Professor Johan Iskandar. Okay, ladies and gentlemen, now we have come to the Q&A or discussion session. At this time, I already have one question from the chat box. Professor, or maybe you wanna have a rest first?
Yeah.
No, okay.
Okay, no problem.
Yeah. The first question is, in the face of climate change issue, what do you think the future of food security or food sovereignty maybe for other indigenous communities in Indonesia aside of Baduy, such as Dayak maybe, or maybe Mentawai? Do you have idea for that?
Okay, thank you for the questions. Based on our experience conducting research in traditional people, there are some traditional agroforestry in many areas of Indonesia, For instance, in Baduy, or in other areas in West Java or southern Banten, it has been known traditional agroforestry called Sweden farming, and also village forest. This is we call agroforestry system, because the land has been planted by many annual and perennial crop mixed together. In other areas in Kalimantan, for instance, there is a special similar traditional agroforestry, which is called as another terms in Baduy is called hutan kampung, leuweung kampung, or huma. But in Kalimantan, that is also traditional agroforestry is called lembo, and also Sweden cultivation that is called uma, and also in other areas is a special traditional agroforestry system. Generally, traditional agroforestry has been managed by traditional ecological knowledge and belief. So the practice of Sweden cultivation or shifting cultivation is based on traditional ecology and beliefs. So in the traditional agroforestry, the land is planted not only planted by annual crop, different from the wet rice farming. Yeah, wet rice farming is usually crop or planted by rice only. But in traditional agroforestry, the land is planted by annual and perennial crop. So if has disaster, yes, for instance, climate change, yeah, long dry time, yeah, and also sometimes there is a long wet season, the rice planting system in wet rice cultivation in sawah, it is very, very serious damage of the rice. But in traditional agroforestry, because the land has been planted by annual and perennial crop, maybe failure of certain species, but can still can be harvested from other species. So also there is input of the agriculture system is very low external input. Yeah, they usually maximize of internal input, organic, fertilizer, biopesticida. So if there's lacks of fertilizer or the fertilizer price is high at the time for the traditional communities, yeah, in many places in Indonesia, is considered not vulnerable for disaster such as the climate change. Based on my opinion and based on my experience, so the monoculture system, commercial monoculture, is not resistant, yeah, to disaster, the climate change, for instance. But the traditional agroforestry can be considered as more resistant to disaster, natural disasters such as the climate change. Yeah.
Okay, thank you. Okay. Thank you very much, Professor Johan, for the explanation. Yes, it's very interesting explanation because as we know that the agroforestry, yes, it provides like how to say more adaptive to the climate change, right, Professor?
Yes.
Okay.
Yes.
Yes. I think as like the time is very limited, Professor. We still want to have more discussion with you. However, I need to summarize our discussion today. Okay. So as I mentioned at the beginning, ethnobiology in Indonesia, of course, because Indonesia has various ethnics. I think more than 500 ethnics in Indonesia, and also we have a lot of like various landscape, and this is provide pathways to enrich agricultural productivity in Indonesia, and again foster the growth of crop varieties and confront food security challenges based on holistic and culturally sensitive and using the sustainable approach. Okay, ladies and gentlemen, now we have reached to the end of this session. So I would like to thank Professor Johan Iskandar for the insightful presentation and explanation, and also thank you very much for the discussion. For all, thank you very much for your kind attention.
Okay, thank you.
Thank you, Dr. Dina Octavia, for your great moderation, and Professor Johan Iskandar for your inspiring presentations. For all participants, the seminar book has been provided by the committee and can be downloaded by the link sent through the comment box.
And also reminder for all the participants, please fill in the attended participants provided by the committee through the comment box. Thank you. Now, we're gonna move to our plenary session 1, featuring 3 distinguished speakers, Dr. Namrata, Professor Ibnu, and Professor Hitoshi. This session was moderated by Mr. Rudy Hassan, PhD. Mr. Rudy Hassan, PhD, has expertise in plant physiology, botany, molecular biology, fiddlecraft, and biology education. For Mr. Rudy Hassan, the time is yours.
Oke. Tes. Tapi belum muncul ya? Udah. Tes. Deh. Okay. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Dear ladies and gentlemen, all attendants of this conference. Let me introduce myself. I'm Rusdi Hasan from Department of Biology, Pajajaran University. Dear ladies and gentlemen, now we are entering the plenary session 1. In this session, we are going to present 3 prominent speakers that I will going to introduce. The first one, Dr. Namrata Tapathamang, as an associate professor from and head of postgraduate department of geology and BBD College that affiliated it in Sikkim University, Tadong, Sikkim, India. Okay, welcome, Dr. Tamang. Welcome to Indonesia. It's honor to us. You can come here as a visit presenter in our conference. Okay. And the second one, we are going to present Professor Dr. Insinyur Ibnu Dewi Buwono, Master of Science. Thank you, Prof. Welcome to this conference. Professor Dr. Ibnu Dewi Buwono is affiliated with Faculty of Fisheries and Marine Sciences in Universitas Pajajaran. Professor Dr. Ibnu Dewi Buwono was born in Malang, 8 December 1982, and education background of Professor Ibnu Dewi Buwono, undergraduate student from Faculty of Fisheries, Brawijaya University. Then from master degrees in Faculty of Natural Sciences, Biology Study Program in Gajah Mada University, and from doctoral program at Agricultural Science in Specialization of Fisheries of Universitas Pajajaran. The third speaker will present the presentation online. It is Professor Hitoshi Iwahashi. Konnichiwa, Professor Hitoshi Iwahashi. Professor Hitoshi Iwahashi was born at Wakayama City, Japan at May 30th of 1958. Professor Hitoshi Iwahashi affiliated with Laboratory of Applied Microbiology, Molecular Life Science, Department of Applied Life Science, Faculty of Applied Biological Science, Gifu University. Okay. Bachelor degree of Professor Hitoshi Iwahashi is at Hokkaido University, as well as a master degree and doctoral degree at Hokkaido University. Yes, welcome for all speaker, prominent speaker kita of our conference will be present each 15 minutes. Okay. So we have 30 minutes for this session. For presentation, each presenter have 15 minutes. So for the 3 presenter, there is allocated time 45 minutes, and then question and answer is 15 minutes. Okay. Okay. We continue to the first presenter, Dr. Namrata Tapathamang. Welcome to Indonesia, to Bandung. Dr. Namrata Tapathamang will present the title of Bacterial Community and Safety of Ethnic Fish Product of India. Yes, please, Dr. Namrata Tapathamang, time is yours.
Hello.
Hello? Yes, it is there only. It's with sir. Sir's, he has already given. Yes, PPT. It's written Namrata. It is written Namrata.
Yeah, I think we already give it back to our husband, and we haven't copied it yet. But it's okay. Don't worry. I think we haven't copied it yet. We will ask
Okay.
Professor Jyoti first, and then Pak bisa ditukar dulu jadi Prof. Hitoshi dulu. Boleh, ini ada sedikit trouble teknis. Udah ready, udah connected ya. So maybe Prof. Hitoshi will go first if that's okay with you, so we can prepare the material.
Yes.
I'm really
No
sorry
problem.
for
No
this.
problem. No, it's all right. No problem.
Okay, yes, yes. Okay. Dear all attendants, I'm sorry for the first presenter would be Professor Hitoshi Iwahashi from Gifu University. Welcome, Professor Hitoshi Iwahashi. Konnichiwa. You are going to turn to the first person in this session with the title Genomic Analysis of Japanese Traditional Fermentation Food for Safety and Sustainability. For Professor Hitoshi Iwahashi, please, time is yours. Does Professor Hitoshi Iwasahi already connected with us? Maybe I don't know. Is he already? Is he already? Okay. Okay, I'm sorry, ladies and gentlemen, Professor Hitoshi still unconnected with us. So the first presenter go back to Dr. Namrata Tapatamang. Please, time is yours, Doctor.
Hello. Hello. A very good morning to all of you. First of all, I would like to give my sincere thank you to Rector, Professor Ian of the university, and I'm also very thankful to Professor Sapitri for inviting me in this beautiful university. My name is Namrata Thapa Tamang. I am from northeastern state of India, Sikkim, working in postgraduate department of zoology, Gantok, Sikkim. So my presentation will be on the topic bacterial community and safety of ethnic fish products of India. So this is the map of India. First of all, I would like to mention that I am not from the core part of India. We are from the northeastern part of India, more towards South Asian countries. That is why many people are confusing me with the people of Indonesia. They are speaking Indonesian with me, but I'm not Indonesian. I'm an core Indian. So this is the Sikkim state where we are living, and I am talking about my state first. So in Sikkim, we have only one river system, that is Tista river system, where we find the plenty of fishes which are mostly indigenous in this nature. So to talk about the fermented or as well as ethnic foods from my state, I would be focusing only on the fish products, because most of the traditional fermented as well as non-fermented food products have been already explained by Professor Tamang. So because my study is mainly focusing on the fish products, so I would be talking only on the fish products of my state, my country. So categorically in our state, there are two different types of fish products available, which are mostly prepared indigenously, or we can say traditionally, by the people of our state, and they are mainly based on fish salt formulation as well as fish salt carbohydrate mixtures. We have many more examples of fish products which are available throughout the world, especially concentrated in the South Asian countries, as well as in our country. It is mostly found in the northeastern part of India. So this is the list of fish products which are available in India. These are all in our indigenous language: Sukakomacha, Nuchi, Sidra, Sukuti, Ngari, Hentak, and Tungtap. And interestingly, some of them are purely dried, some of them are sun-dried, some of them are fermented, some of them are semi-fermented, and so on. So we can get the varieties of fish products in our state. Traditionally, all these fish products are the most important dietary identity of the Northeast people, because throughout India, you may not find these kind of fish products, because we have our own culture, ethnicity, and our tradition is different because of the it's a rich biodiversity of our state, Sikkim. We have different types of food culture system where we produce different types of fish products ethnically by our own people. So we have elaborately worked on the physicochemical analysis of the fish products as well as meat products which are available in Sikkim. We have analyzed the pH value as well as moisture content of the fish products as well as meat products, and we have found that in most of these products, pH as well as moisture have been more which are found in the high altitudinal areas of Sikkim. And this is the flow sheet by which the various different types of fish products are prepared by the people of India, or in my state, Sikkim. And this is also a picture showing different types of fish products and their mode of consumption by the people. and you can see one of these, some of them also have medicinal values where the people consume these fish products when they are sick. These are also many more, and interestingly, some of them are more identically similar to the fish products of Malaysia, Thailand, and Indonesia. Of course, the way of preparation is slightly different. And we have done some advanced level research work on these fish products, like high throughput sequencing, as well as and also produce the paper. And some this high throughput sequencing studies have shown the variety of microbial diversity which are found in these type of fish products, as well as this is also a graph where we have studied the different species of the microorganisms which are found in the different types of fish products. And also this is also a heat map showing some statistically significant and some very rare species of fish microorganisms like Cytobacter, as well as which is available in these fish products by the high throughput sequencing mechanisms. And this is a diagram showing the predictive microbial functions on the basis of against the KEGG protocol, which is mainly based on the ecosystem of the microorganism, their human diseases, as well as their functional prediction and all. So all the like parameters have taken, and the study has been done on the basis of KEGG system. So highlighting this research study, we have profiled nearly 201 species of the bacteria, and among them one of the most important and abundant species were found is Cycobacter celer, supposed to be one of the very important bacteria, mostly present in almost all the fish products of Sikkim. And we have also discovered one very important and very noble genus of bacteria, that is Rumelibacillus, which was mainly found in one of the fish products, Sidra. And we have done this study on the basis of the clustering and scattering pattern of the bacterial compositions, which showed the diverse population of bacteria in these fish products. So, and not only that, when we did the predictive functionality of these samples, we have discovered the remarkable predictive functionality in which microbiota, which has shown the very important microbiota functionality relationship among themselves. So these are some of the papers that we have produced in our research times, though I do not have much time to explain all these, but I have given a very short informations about all the research activities that we have performed in our small lab, and I have also produced one of these paper. And this is my core team. We work in Sikkim NBBD College, Tadung, and this is these are my research scholars who have done the work on this. Thank you very much.
Well, thank you, Dr. Tamang. Okay, the excellent presentation about how to present bacterial community and safety of ethnic fish product in India. It's explained that identified more than 200 species of bacteria in local research in India. So it's very, very good, very excellent finding to improve the local fish and how to preserve the fish in India, especially. Okay, thank you, Dr. Tamang. Ladies and gentlemen, please keep your question. We will go to the next presenter, Professor Professor Dr. Ibnu Dewi Buwono will present the presentation with the title Transgenesis GH or growth hormone to improve growth characteristics and reproduction of catfish strain in Indonesia. Okay, thank you, Professor. Okay, welcome to this conference. Okay, time is yours.
Good morning, ladies and gentlemen. Thank you for the participation. Today I want to sharing my research about transgenesis growth hormone to improve growth characteristic and reproduction characters of catfish strain in Indonesia. Problem: catfish aquaculture in Indonesia has been around for almost 20 years in decline genetic qualities. The intensive culture has been used the catfish strain of Dumbo and Sangkuriang. The spawning causes a decrease in a growth and reproduction characteristic, especially male fish experience a decrease in gonad fertility when dry session male gonad maturity cycle is low. Insertion the gene encoding growth hormone in related fish can induce the propagation of male gamete cell of sperm and gamete cell female of eggs, improving the fertility of male fish in the dry season and increase a number fertilized eggs. Ultimately can increase the production of dry season catfish larvae and fingerling. Next. The development local catfish aquaculture is not encouraged, considering that they grow slowly and susceptible to pathogen, especially Aromonas hydrophila bacterial infection. In order to respond this conduction, the Directorate General of Fisheries introduced catfish from Africa, Clarias caripinus, which has a relative fast growth rate and resistant to Aromonas hydrophila in 1985, known as Dumbo strain of catfish. As a superior catfish, the Dumbo strain from catfish culture in Indonesia decreased genetic quality after 25 years of African catfish culture. There is development of several new strains. 2004, the Sangkuriang strain was formed. 2014, the Mutiara strain was formed. And 2016, the Mutiara transgenic growth hormone strain was formed. Breeding transgenic growth hormone Mutiara catfish, GGGH inheritance of G1 42%, G2 50%, G3 70%, G4 74%, G5 92% increase more stable. Next. Different in the gain encoding growth hormone for Dumbo versus Mutiara strain of catfish. Alignment of growth hormone Dumbo and Mutiara sequence with bioedit version 7.0. The same sequence consensus of 53% indicated that but catfish is a different strain. Blast X analysis, nucleotide alignment of Mutiara growth hormone and gene bank show sequence similarity 65% indicating that the sequence growth hormone Dumbo and Mutiara are heterolog. Next. Development of catfish hatcheries in Indonesia. Breeding level. Demand for catfish fingerling in relatively high. Limited water resources. Dry season. Source of catfish broxtooth. Require adoption of breeding technology. superior catfish strain, breeding system and program. Target superior growth stability in each generation can be established. Next. Superior growth in transgenic growth hormone of Mutiara catfish. Current research, growth hormone transgenesis is able to increase growth significantly. Co-salmon 10 times more endophilin, mudslot 2 times NAM et al. Tilapia 2 until 7 times Kobayashi. The growth of transgenic growth hormone catfish was 2 until 3 times higher than non-transgenic catfish or overgrowth. One method transgenesis, sperm electroporation. Next. The gene transfer in the gene encoding African catfish growth hormone, Clarias caripionus growth hormone, GGGH registered in the gene bank accession number MN 24923.8.1 integrated in the PCMV GGGH expression factor 6285 base pair and transfer into Mutiara catfish sperm. Transmission of GGGH G1 42.85%. Transmission GGGH G2 50%. Transmission GGGH G3 74%. Transmission GGGH G4 80%. Transmission GGGH G5 92% become stable growth and become mass production. Next. Application of transgenesis. Map of research protocol. Step 1, insertion of GGGH using electroporation method. Production of G0 transgenic Mutiara catfish. Evaluate the success of transgenesis method. Step 2, spawning transgenic G0 mating non-transgenic G0. Production of G1 transgenic Mutiara catfish. Analysis growth hormone and IGF-1 expression level, steroid level, and gonad histology. Step 3, crossbreed G1 transgenic Mutiara catfish proxtooth. Production of G2 transgenic Mutiara catfish. G1 transgenic fish growth test and fish body protein level. Evaluation of reproduction characteristic. Become production G3 transgenic catfish, G4 and G5. Next. Growth characteristic of transgenic Mutiara catfish as superior fish. Comparison of breeding methods. Methods selection, strain name Mutiara catfish, increase growth 10 to 40%. Method G, growth hormone transgenesis, strain name transgenic Mutiara catfish G1 to G4, increase growth 100 to 100%. The growth of Mutiara transgenic and non-transgenic are shown in this table. Mostly in the increased growth from non-transgenic can be 2 or 3 times. This will be high potential to increase catfish culture production. Next. Exogenous growth hormone expression level. G4 transgenic Mutiara catfish rear at high stocking density. Relative expression of GGH in transgenic G4 catfish 1.5 to 3 times higher than non-transgenic fish. Overexpression of GGH in G4 fish transgenic, increased growth, cause effect of adding the GGH gene Next. Propagation of transgenic mutiara catfish, transgenic catfish line, reproduction character, or breeding scheme. In 2016 until 2017, production of transgenic G0 and non-transgenic G0 mutiara catfish. In 2017 until ya. Sebelumnya. Ya. Nah, in 2021, I'm sorry. In 2016 until 2017, production of transgenic G0 and non-transgenic G0 mutiara catfish. In 2017 until 2018, production and propagation of transgenic G1 mutiara fish. In 2019, production and propagation of transgenic G2 mutiara fish. Next. Continue propagation of transgenic fish line. In 2019 of 2020, production and propagation of transgenic G3 mutiara catfish. In 2021, production and propagation transgenic G4 mutiara catfish. Reproduction characteristic of transgenic catfish. Reproduction characteristic of transgenic mutiara catfish is higher than non-transgenic fish. The average diameter of transgenic fish is higher than non-transgenic fish. The total sperm density per milliliter of transgenic fish is higher than non-transgenic fish. The degree of fertilization and hatching of transgenic fish is higher than non-transgenic fish. Next. Gonad of transgenic and non-transgenic mutiara catfish. The gonad size of female and male transgenic fish is larger than non-transgenic fish. Next. Histology of female and male gonad transgenic fish. The ovarian follicle of transgenic mutiara catfish were observed to be larger than non-transgenic female catfish. The level of maturity of the female gonad of transgenic mutiara catfish is in the mature category is indicated by formation the yolk globule, granulosa cell, and theca cell. Like this, the testis transgenic in development faster than non-transgenic fish, which is characterized by formation of spermatozoa abundant than non-transgenic spermatid and secondary spermatocyte. Next. Amino acid and protein content of transgenic catfish. Transgenic fish products as food are aimed to fulfill people's protein consumption need, considering that Indonesian population continues to increase and availability of food protein from fish cannot be met at any time. Effort to meet this food need have encouraged various research including genetic engineering technology or transgenesis to produce superior fish with higher protein than non-transgenic. Protein content of transgenic mutiara catfish G1 to G4 mean 18.92%, G1 21.182%, G2 71.61%, G3 and 19.49%, G4 higher than non-transgenic fish. Next. The meat test of G3 and G4 transgenic catfish. The meat taste, aroma, texture, or stage of G3 transgenic Mutiara catfish so that respondent rate transgenic fish product sample as more preferable and then non-transgenic fish sample. The organoleptic test of G4 transgenic catfish are better than non-transgenic catfish. The meat quality of transgenic Mutiara catfish as food and popular with consumer in compensated for high protein content in transgenic catfish compared to non-transgenic fish, indicating that increased protein level in G3 and G4 transgenic fish contribute to improving the taste, smell, and texture to the meat. Next. Next. Allergicity test. Histamine level in G4 transgenic catfish meet the results, so the histamine level transgenic fish is under 3 milligram per 100 gram. This result is lower than non-transgenic, 3.5 milligram 100 gram, but are still under the minimum limit permit by Food Drug Administration or FDA, 5 milligram per 100 gram. Next. Detection of CMV residue 205 whisper amplicon in G4 transgenic fish. Detection of PCMV, cytomegalovirus promoter sequence. No GMV promoter was found in the G4 transgenic fish. PCMV, not coding gen sequence and non-viral sequence. And transgenic Mutiara catfish as safety food for consumption of human. Next. GGGH registration submitted to GenBank. The size of GGGH amplicon is 615 base pair. The nucleotide sequence 1 to 615, which is translated into amino acid, is the growth hormone protein African catfish or Dumbo stream. Next. This is recent publication about transgenic Mutiara catfish. One is about growth hormone transgenesis and feed consumption influence growth and protein and amino acid content in transgenic G3 Mutiara catfish. Another one is about the growth and expression level of growth hormone in transgenic Mutiara catfish, second generation. Next. This is about GGG8 insertion functional domain analysis in transgenic G1, G2, G3 Mutiara catfish broadstock. It showed that functional domain of fish growth hormone protein are conserved in G1, G2, and G3 and indicate stable exogenous growth hormone inheritance to produce transgenic catfish strain in each generation. Thank you so much for your interest and attention.
Well, thank you, Professor, for a great presentation about transgenic for catfish. As we know that catfish is very popular in Indonesia, Professor. So it can be improved by a transgenesis method, so not only for growing the catfish, but also it's a very proposed to be consumed for Indonesians. So it's safety protein content also higher than control, Professor. Okay, thank you, Professor. Okay, ladies and gentlemen, please keep your equation for Professor Ibnu. Then we finally come to the third speaker for this session. Is Professor Hitoshi Iwahashi already online? Hello. Okay, okay. Konnichiwa, Professor Hitoshi Wahashi. Welcome to this conference. So this time, your turn to present your presentation. Please make a presentation. I remind that you have 15 minutes to present the article you conducted with the title "The Genomic Analysis of Japanese Traditional Fermentation Food for Safety and Sustainability." Okay, Professor Hitoshi Iwashi, please, time is yours. Okay, okay. Please wait. Yes. Okay, Professor Iwashi, do you want to share it by yourself or we? Okay, 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 thank you. Please. Yes, I do. Yes, yes, we do it.
I try to